Yeah, my dear students, what's up? How do you do? I think everybody doing well, right? Done. Yeah, I am well. Thank you. Thank you very much. Myself, Mr. Karthikeyan, mentor in NEET, Boskin and Zoology subjects. Welcome to my video session. In this video module, I would like to explain to you the structure of monocot stem, TS, the anatomical tissues arrangement. A clear diagrammatic presentation is on the board. Then one by one I shall explain to you the entire arrangement. Try to understand. One of the essay answer question, 8 marks it carries, NCERT syllabus, and MSET and NEET bits also can be raised from this topic. Let's see what type of bits will be raised. Before that, the clear information, we will be started. Good luck. If we observe the main frame or structure of monocotyledonous stem, the main parts are nothing but epidermis, cortex, vascular bundle, nothing but a steel. Epidermis, cortex, steel. So these three main parts are located or arranged in the form of tissue system in the monocot stem transverse section. Let's see one by one. Number one, epidermis. So many times I have told you what is meant by epidermis, nothing but epi means upon, dermis means layer. So it is the outermost layer. The main function is protection of innermost tissue in the stem. The epidermis is a single layer structure. Cells are closely, tightly, compactly arranged. No intercellular space or gaps. On the epidermis, there is a thick waxy coat that is known as cuticle. So the cuticle is thick. If we compare to dicot stem, monocot stem cuticle is thicker. Then I have told you already the main function is protection. Stomata are present, here and there stomata are present, they helps in the gases exchange, directly indirectly transpiration process. Followed by cortex. So here cortex in the sense, in dicot stem and monocot root, dicot root, cortex is subdivided into exodermis or outer cortex or hypodermis, then general cortex, then endodermis. Innermost layer of the cortex is transformed or modified into endodermis. But coming to monocot stem, a small change in the tissue arrangement that the cortex, majority of the cortex is made up of only parenchyma, that is ground tissue. So here endodermis is absent, pericycle is also absent. Dear students, be careful, these all points. So one by one we will discuss, then you can get a clarity. Followed by epidermis, 3 to 4 layers arrangement, the tissue is known as hypodermis. Here in monocot stem, hypodermis is made up of sclerenchyma. In dicot stem, hypodermis is made up of collenchyma, but here the tissue is sclerenchyma. The main function is giving mechanical strength and rigidity and support to the stem. And one more characteristic feature is in the hypodermis, here and there parenchymatous tissue is found. This parenchymatous tissue, it has chloroplasts, because the parenchyma which possess chloroplast is known as chlorenchyma. Remember. So here chlorenchyma, a type of parenchyma, can be seen. It will be participating in the photosynthesis. Then followed by cortex. So this is known as general cortex or else ground tissue. Why? Because huge number of cells are arranged, many layers of cells are arranged. It is also known as ground tissue. Why? Because generally plant body is made up of more than 70% of parenchymatous tissue. Because cell is the basic unit of life, right? Same like parenchyma tissue is the main life for the plant body, because more than 70% it occupies in the plant body, the entire place, whatever. So because this is known as ground tissue or basic tissue. Remember, huge intercellular space is present, thin cell wall, because parenchyma, this is main function is storage, right? Then coming to next part. So here endodermis is absent, pericycle is also absent. Then what these structures are? The structures scattered in the ground tissue or parenchyma, each and every one is known as vascular bundle. So directly, indirectly we won't call this as steel. Why we won't call like steel, sir? In dicot stem and monocot dicot roots we are calling steel. But why we are not calling here as steel? In the sense, there is no endodermis, because this is not 100% will be considered as steel. Just vascular bundles, conductive tissue, transporting tissue, complex tissue. Because once again I am repeating, endodermis is absent, pericycle also is absent. Only xylem and phloem is present. And this xylem and phloem is surrounded by, enveloped by sclerenchymatous tissue layer, is known as bundle sheath. So vascular bundles which are present in the monocot stem, they are surrounded by sclerenchymatous bundle sheath. Here it is not called as pericycle or endodermis. Remember, these are the main points. Okay, done. Then if we observe keenly, clearly, carefully, this is xylem. These both are known as metaxylem, two large vessels, and here we could observe protoxylem, small xylem vessels, right? This metaxylem and protoxylem arrangement looks like roughly Y shape. So the arrangement of vascular bundles, especially xylem, in the monocot stem is roughly Y shape. Remember, Y shape. Then xylem, we could observe xylem followed by the, we could observe phloem followed by the xylem. So between phloem and xylem there is no extra tissue, nothing but a cambium. But in a dicot stem vascular bundle, we have been observed between xylem and phloem, cambium or cambial tissue is present, because that is conjoined collateral open. But here this vascular arrangement is conjoined collateral closed. Why? Because cambium is absent. Remember everybody. Okay, then coming to protoxylem, exactly near the protoxylem there is special structure. The structure is known as lysigenous cavity. So what is this lysigenous cavity? Nothing but this cavity can be stored water. So some of the protoxylem vessels, they are degenerated, disintegrated, then they are converted or transformed or modified into lysigenous cavity to store the water. Main competitive bit, remember. Then one more special feature, around this are close to protoxylem, xylem parenchyma tissue is located. Look here. Xylem parenchyma tissue. Protoxylem, lysigenous cavity, xylem parenchyma. And in this xylem parenchyma, here and there, xylem tracheids also found. Here we did not show in the diagram, but competitive bit very important. Xylem tracheids also found around this protoxylem in the xylem parenchyma. My dear students, please try to understand. Then, due to absence of what it is called pericycle here at cambium, we are unable to observe secondary growth. In dicot stem, dicot root, we could observe secondary growth, but in monocot stem, we are unable to observe secondary growth. And one more thing, try to compare these two vascular bundles with this. Any change? Could you observe? Yeah, size. This one is larger or bigger in size. These both are smaller in size than this, right? So, towards peripheral, the vascular bundles more in number, small in size. Towards inner side, the vascular bundles are bigger and larger and less number. This is one of the main point for competitive purpose. Towards peripheral, large number of small sized vascular bundles are located, and oppositely towards inner, less number of large sized vascular bundles are located. Please take care. And these vascular bundles are scattered here and there, scattered in the ground tissue or the cortex, because this type of arrangement, vascular bundles arrangement, is known as atactostele. Remember everybody, atactostele. But in dicot stem, these vascular bundles are aligned in the circular pattern, ring pattern, particular ring pattern. So such type of arrangement is known as U-stele. So this is atactostele, that one is U-stele. So my dear students, I hope everybody understood. So once again, final briefing. Thick cuticle, epidermis, hypodermis, sclerenchymatous tissue, chlorenchyma, a type of parenchyma, photosynthesis, followed by ground tissue or general cortex, huge intercellular space has been found. Then main function is storage. So here vascular bundles are scattered. Pericycle endodermis is completely absent. Secondary growth is also absent. Y-shaped arrangement xylem. Then extra special structure lysigenous cavity. Xylem fibers and tracheids can be seen. Bundle sheath cells are made up of sclerenchyma. Then this arrangement is called atactostele. Towards peripheral, more number of vascular bundles, small size. Towards inner surface or towards inner side, less number of large sized vascular bundles are found. Yeah, I hope everybody you are understand well. In next video, I shall meet you with one more beautiful topic with clear explanation. Thank you. Signing off. This is Mr. Karthikeyan, mentor in bone skin and zoology subject.